So just to welcome everybody on behalf of the Libman Institute and the Department of Cardiac Sciences and Physiology and Pharmacology, we're very proud to have shortlisted Dr. Wang for a CRC Tier 2 chair position in Immuno-Cardiology, which we're offering a position. Dr. Wang, to give you some background, is a visiting instructor of the Division of Vascular Surgery at Stanford University. She obtained her PhD training in basic sciences at the University of British Columbia, followed by working with physician scientists at the Center for Heart-Lung Innovation at St. Paul's Hospital and Stanford University. She's a hybrid vascular biologist and pharmacologist. Her research goal is to develop new therapeutics and diagnostic tools for better treatment and prediction of atherosclerotic disease. Her recent publication revealed the underappreciated roles of vascular smooth muscle cells in atherogenesis, which challenged the traditional dogma that atherosclerosis is purely a macrophage-driven disease. She has pioneered a number of innovative techniques in lineage tracing and high-throughput cell profiling in the context of blood vessels. Her research was recognized by the Early Career Investigator Award and Fellowship from the American Heart Association and the Duke Junior Investigator Research Fund. She aims to push forward the limit of multiplex in situ single cell profiling to unveil the functions of diverse cell types in human atherosclerotic lesions. She'll present her talk today entitled Targeting at the Hidden Gems, Smooth Muscle Cells in Atherosclerotic Disease. So welcome, Dr. Yang. Take your time. We've scheduled an entire hour for this. We were expecting sort of a 30 or 40-minute talk, so we can still get your talk completed with some short period of question and answers. So thanks so much for logging in, and please go ahead.
Sure, and thank you so much for the introduction. So can everyone see the slides?
Yep.
Okay, thank you very much for the introduction. It's a great pleasure to get connected with you remotely from this difficult time. I actually originally come from Wuhan, so really I want to urge everybody to stay physical distance and stay healthy to protect yourself and people around you. So today I want to actually start talking about what does the word diaphragm. It's a very hot topic recently because everybody is panicked about the pandemic of COVID-19. But I want to remind everybody that I don't think that the COVID-19 pandemic outbreak is going to change the landscape. That cardiovascular disease shown here is still the leading cause of death worldwide, contributing for 17 million of deaths every year. And most of the cardiovascular disease is caused by atherosclerosis, which is the buildup of fatty streak on the blood vessel that narrows the lumen. This ultimately trigger cardiovascular events like heart attack and also a stroke. So despite that, cardiovascular disease is the leading cause worldwide. Therapeutic design seems to have to be slowing down in the recent 20 years. As you see, the FDA-approved drug has been decreasing in this category, whereas cancer drug is increasing and doing really well. So I happen to work in industry on cancer drug therapeutic development for 1 year. So by comparing both areas, I kind of formulated my own opinion about why this is happening, why the area in cardiovascular disease has been slowing down. Is that I think that a lot of the focus recently have been really focusing on a few conventional risk factors that trigger cardiovascular disease, such as lipids. It's a great drug to target at lowering the lipid to treat cardiovascular disease. But however, as is calculated there, even though when we were able to lower down the LDL level to normal, there is still a substantial residual risk contributing to cardiovascular disease that account for 70% of the total risk. So the conventional risk factors do not account for all. And for me, that I think one of the unmet needs to address the issue of cardiovascular therapeutic development is that we need to open up more avenues to design therapies to target the residual risks, understand what are the residual risks and how do we treat them. The second thing is that once we define this residual risk, we obviously need biomarkers to identify and assess this risk. This is critically important as I see that we used to look at patients with cardiovascular disease as more of a homogeneous population that carrying high cholesterol levels. Whereas in cancer, we really phenotype the patient based on what risk they carry, and then to identify who will be the beneficial patients and to evaluate the drug effects. So having said that, these are the two unmet needs that I think. Next, I will show you that how my previous research, the two postdoc research done at UBC and Stanford, identify what I think are the residual risk factors and how do I treat them. And also, how do I want to leverage this experience to address the unmet needs that's in my future lab. So what are the hidden risk factors? So if I show you this busy converted Manhattan plot, showing you the genes that pop out that's associated with coronary artery disease, actually a lot of the genes are not related to cholesterol metabolism. Instead, a lot of the new hits popping out that's related to cell fate, like cell differentiation, and also like atherosclerosis. A lot of these cell fate and cell biology related risk factors reside in a cell that's intrinsic to the blood vessel wall, which is smooth muscle cell. So smooth muscle cell forms the medial layer of the blood vessels, and when it's in the healthy differentiated stage, it's of a contractile type that express a lot of the contractile protein like smooth muscle cell alpha actin that we always use to probe smooth muscle cell in the conventional immunohistology method. However, during atherogenesis, it converted to a synthetic phenotype that it did differentiate to lose all of the differentiation markers. Here, I show you the first smooth muscle cell lineage tracing model done by Falk's group. Giving you here, this is an atherosclerotic plaque. You see there's a substantial amount of blue smooth muscle cell within the plaque, but less than 40% of them still express the differentiation marker smooth muscle cell alpha actin. So it means that smooth muscle cell has been hidden there in the plaque without knowing it because we have been using the traditional method to probe for them, but they have already de-differentiated. And having said that, instead it found that this de-differentiated smooth muscle cell actually pick up a lot of the marker that's being thought to be unique for macrophages instead. So this suggests that a lot of the smooth muscle cell may have been misidentified as macrophage before. So what I think that in the next step at UBC, I'm really interested in studying how do these differentiated smooth muscle cell contribute to atherogenesis. So I think if smooth muscle cell can convert to macrophage, and one of the jobs that macrophage does in the plaque is to form foam cells, then maybe the smooth muscle cell can also take up the job to become smooth muscle cell foam cell and thereby contribute to atherogenesis. This picture was supposed to show you that how hard it is to count and identify individual foam cells in the lesion, especially when the cells are touching each other and squished. It's really hard to count. So these are the excuses that I used when I was a postdoc to complain about how tedious it is to use conventional method to count foam cells to characterize them in the lesion. Somehow this picture was picked up by the editor of ATVB to always remind me the story that if you will complain about conventional method, then you really need to face the challenge to establish a new shortcut for yourself and prove that it's right. So I went on and established a flow cytometry-based method to count and characterize foam cells. What I did is that I digested the aorta to single-cell suspension and run millions of cells through the flow cytometry to characterize it in a few minutes. So I used BODP to identify foam cell based on their intracellular lipid level, and I use a pan-leukocyte CD45 to isolate all the macrophage-derived foam cells. And then the remaining foam cell, which is CD45 negative, I will need to confirm that whether they are from smooth muscle cell lineage or others. So the next step is that I sort this CD45 negative foam cell, and then I did a confirmation of a lineage test by an assay invented at Gary Owen's lab. So this is the world's first and only so far epigenetic mark that has been found that's persistent even during smooth muscle cell transdifferentiation. So with this epigenetic mark unique to smooth muscle cell, I found that this mark is only enriched in the CD45 negative form cell population, confirmed that the majority of the CD45 negative form cell are of a smooth muscle cell origin. Having confirmed their lineage, I went on to use the flow cytometry method to count how many foam cells are from them. And surprisingly, I found that even in female and male apple knockout mice, there are actually 70% of total foam cell population are smooth muscle cell derived. They actually hold half of the cholesterol in the plaque. I was really surprised by the result because I saw that all the foam cells are mainly coming from macrophage myself. And these smooth muscle cells are a really hidden reservoir of cholesterol. Then I think, that does it matter? Even though they are different cells, but they are doing the same job. So can we just treat them as the same? Then when I went deeply into study the biology of the two types of foam cell, I found that the macrophage foam cell is more of a professional job taker to take out the lipids. So when it forms foam cell, it's very professional that it upregulates ABCA1, which is a cholesterol exporter that's been lining there to help this macrophage to get rid of the intracellular lipids through a process called cholesterol efflux. So this means that it's actually beneficial once you trap the lipids in the macrophage, you can also get them out from the blood vessels through this process if they can upregulate ABCA1. However, when you look at smooth muscle cell foam cell, it's been very lazy to upregulate ABCA1 when they become foam cells. And also they are very reluctant to give out the lipids once it's trapped there. So what does it mean? It means that about half of the cholesterol in the plaque has been trapped in the non-professional foam cell, which are very reluctant to give out the lipids later. This has been also indicated by an editorial review done by Gary Owen along this paper by appreciating that how smooth muscle cell contribute to atherosclerosis is that it's simply the wrong cell take up the wrong job and then trap the lipids in the blood vessel. In this case, it really contributes to the lipid deposit in the blood vessel. So having said that, I have shown you that smooth muscle cell is a hidden reservoir of cholesterol, and it contributes to 70% of total foam cell formation. Now, because it is resistant to cholesterol efflux, a lot of the lab, instead of targeting at the ABCA1 mechanism, they're targeting at how to manipulate the metabolism of smooth muscle cell to have them deposit less to become less foam cell-like. So of course, I'm very proud that a lot of the labs now are using the flow cytometry method that have established to characterize foam cells. So now you're still possibly trying to digest and being very surprised about statement that smooth muscle cell contributes so much to foam cell formation. And I was also very shocked by the result at that time because I think that there are not so many smooth muscle cells in the intima anyway. Then how do they get there? How do they proliferate in the intima? So I went back and to look at Russell Ross' response to injury theory. Actually, he appreciated that proliferation of smooth muscle cell is a signature of atherosclerosis. And there I found a very old paper from the Band-Aid group even proposed that the entire human plaque you have seen is coming from the proliferation of only a few smooth muscle cells, and they undergo clonal proliferation. This makes them look like a tumor cell growing on the blood vessel. So I got really interested into this concept. I think that atherosclerosis is a proliferative disorder of vascular smooth muscle cells. And next step at Stanford, I'm interested in studying how do those smooth muscle cells proliferate in the plaque. Of course, to prove Band-Aid theory about clonal expansion of smooth muscle cell in the plaque, there are two problems I need to solve. First thing is that I need to lineage trace the dedifferentiated smooth muscle cell in the plaque. The other thing, I need to track the clonal origin of this cell at the same time. So by collaborating with Irving Weisman, I have this beautiful rainbow construct crossbreed with apple-called mice. As you can see here, under normal condition, all the cell will express the GFP and become green. But when I give the tamoxifen will activate the Cre enzyme only in the smooth muscle cell. This Cre enzyme will choose any of the arrow with a pairing color over here to cleave at the reporter genes. This results in the removal of the GFP, and all the smooth muscle cell will switch from green to either of a yellow or blue or red cells. And this selection is purely random. So what it does is that by activating Cre only in the smooth muscle cell, we finish smooth muscle cell lineage tracing. And also by randomly select the colors to provide these gene editing events, all the mother cell and their descendant cell will from now on express the same colors. This means that if you see a patch of smooth muscle cell expressing the same color together, very likely they are from the same clone. If you look at the rainbow mice in the healthy media, it's more of a mosaic pattern of all the colors suggests that all these cells come from a different clonal arranging. But as the plaques just start to build up, you start to see one smooth muscle cell clone trying to take over the entire plaque eventually that form the entire lesions. So what drives this smooth muscle cell being so crazy to undergo clonal expansion? There has to be some mechanism to explain it. And at Stanford, the way that I learned is that you can really use a hypothesis-free high throughput method to study the mechanism. So what I did is that I used the single-cell RNA sequencing, dissociate the plaque into single-cell suspension, and isolate this plaque smooth muscle cell to run and to look at their RNA profile. So based on the result, smooth muscle cell is actually very heterogeneous within the plaque. At least we found the 6 clusters of different phenotype of smooth muscle cell in the plaque. And one of them is especially interesting in that it highly upregulates SCAL1, which is a stem cell marker. So this stem-like smooth muscle cell I found is very rare in normal condition, but it's highly enriched when the plaque starts to grow, and it's also enriched in the dominant smooth muscle cell clone, suggesting that it's doing something with clonal expansion. Then right now here, before we move on, I actually told my colleague, like, wait, we have to stop to do a reality check because you know what? I didn't find SCAL1 expressing in human. So we can spend years studying atherosclerosis in mouse with these stem-like cells, but there's no SCAL1 expressing in human stem cell. How do we translate what we learned in mouse to human? We need to do the reality check first. So we went on and collaborated with a bioinformatic expert, Johan. So he has a powerful model called StarNet co-expression module. So with this co-expression module, he actually compared transcriptome profile between our SCAL1 smooth muscle cell and also in human aortas. And he subtracts similar transcriptome profile of a cell cluster that's being shown over here. And this is confirmed that this is the human equivalent SCAL1 smooth muscle cell. And the expression of this smooth muscle cell has been highly enriched in human aorta and also is correlated with risk score of a cardiovascular disease. So here, we are actually very happy that we found the smooth muscle cell equivalent stem cell in human. And then now we can keep going to say how this SCAL1 smooth muscle cell drive clonal expansion. So from here, we found that in this cluster, the most upregulated gene is complement component C3, which is a part of the immune system in the complement pathway. I will not try to pretend that I'm an expert in immunology, but so far what I learned is that by pumping out C3, and this C3 is actually breaking down into C3A, which we found that C3A can stimulate smooth muscle cell proliferation, driving clonal expansion. But on the other side, it also breaking down into C3B, which then later is formulating an 8-me molecule IC3B that decorate the apoptotic smooth muscle cell. In this step, it marks the cell to attract the phagocytes to eat this apoptotic smooth muscle cell by a process called atherosclerosis. So the atherosclerosis, I want to spend some time to explain it. It's actually from a Latin word, and it means burying of the dead bodies. So it's supposed to be a beneficial process that happens in the atherosclerotic lesion. So before the apoptotic cells start to burst and then leaving harmful content to the plaque, there are also macrophages around it trying to eat that apoptotic smooth muscle cell. In this way, before it bursts and causing inflammation or secondary necrosis, you can really help to remove this cell properly. And this is a phenomenon that you can frequently see in the plaque as well. It's a beneficial process that we think by doing this, the C3b arm by atherosclerosis is trying to balance out the proliferation arm the C3 generate, which cause proliferation. But however, as we see that in the plaque, while most of the macrophages are of an inflamed M1 phenotype, these M1 macrophages cannot recognize the 8-me molecule IC3B, and thereby you diminish the pro-atherosclerosis arm of the C3 and only leaving the proliferation arm of C3. And that's how I think SCAL1 positive smooth muscle cell accelerate clonal expansion. Then the next question becomes, if that's the case, then how can we treat this disease smooth muscle cell that has been aberrantly proliferate? How do we bury them? Actually, atherosclerosis is not just regulated by the 8-me molecule. It's a complex system full of the don't eat me and 8-me signals that's between the macrophage and the target apoptotic cells. And actually, one of the don't eat me molecule has drawn my attention. So this molecule is called CD47. It is the don't eat me molecule that's been highly upregulated on cancer stem cells. And now there is a company called CD47 that has generated antibody, and by blocking the don't eat me signals, it's very helpful for them to remove the tumor cells in their test. And this antibody has been in clinical trial right now, in which we have been working with them. So in this case, I'm thinking that the smooth muscle cell that I have seen is having the same similar biology with the cancer stem cell, in which both of them undergoes aberrant proliferation. Then can I use the anti-CD47 antibody in a way to treat atherosclerosis just like a cancer cells that's been growing on the blood vessel? I went on to give this antibody to my rainbow mice, and in the control group, you see the massive clonal expansion of smooth muscle cells. But after antibody treatment, the most significant differences you see is that you lose these big smooth muscle cell clones in the deep intima. I went on to do the statistic analysis. It's showing you that in detail when you just look at the lesion. So the antibody treatment reduced the lesion cells dramatically. And this effect is somehow selective in that it doesn't reduce the lesions, the non-smooth muscle cell portion, but it has been very specific about the lesion smooth muscle cell. And also, if I dissect the structure by only looking at the cap region, well, you always need the smooth muscle cell to be invested there because it helps stabilize the lesion, that you don't want to lose this smooth muscle cell. Actually, the antibody didn't remove smooth muscle cell in this region, suggesting that it's somehow doing the magic of only cropping out the diseased smooth muscle cell line under the intima, while most of the inflammation is happening over here. So with this, I have shown you that aberrant proliferation of smooth muscle cell is a residual risk factor contribute to atherogenesis. It can escape from atherosclerosis due to a defect in the M1 macrophages not be able to recognize the 8-me molecule. But amazingly, since it has a feature that's similar to cancer, we actually found that anti-CD47 antibody, anti-cancer drug, that has been very efficient to remove the diseased smooth muscle cell. Actually, the phenomena of a smooth muscle cell clonal expansion has been appreciated by more study focusing on cardiovascular disease. And there is paper published, which I wrote an editorial on, that also confirmed that clonal expansion of smooth muscle cell is also causal to aneurysm disease. The next step in the LEAPR lab will be how to leverage the anti-CD47, but also in a pharmaceutical format in which we capsulate in a nanoparticle and hopefully provide some targeted pro-afrocytosis effect. Now, from my 2 previous studies, I hope I have convinced you that I think diseased smooth muscle cell is a residual risk factor for atherogenesis. It does it in the way that it forms foam cells that form a dead cholesterol pool that's really hard to remove the lipid out from the blood vessel. It does that by aberrant proliferation like a tumor cell growing on the blood vessel. And all these bad things has been happening without notice. It's being hidden there because they undergo phenotypic changes that we couldn't detect them anymore. And having said that, my future to address the 2 unmet needs I've mentioned before and to target at this residual risk factor, there are 2 major blocks of my future study. So one is to design therapy targeted at the diseased smooth muscle cell. And more specifically, in my near goal, I was really interested in continuous study about how this aberrant proliferation has been happening in the diseased smooth muscle cell. The other part is about develop biomarkers to identify diseased smooth muscle cell. And because they undergo dedifferentiation process, it's been really hard to extrapolate what we've learned in mice to human because we definitely can't do Cre-Lox-P gene knocking and knockout within the human part. And we need to develop markers to identify the human smooth muscle cell. With these 2 near goals, I formulated a 5-year plan into 2 directions. And one is to determine the epigenetic changes that drive proliferation of smooth muscle cell, and the other one is to define the protein marker to identify human smooth muscle cell. So for the first direction, I've shown that in athero, there is smooth muscle cell stem cell undergo clonal proliferation. It's highly heterogeneous. These are the features that's shared by cancer stem cell, in which you have cancer stem cell undergoes initial somatic mutation that kicks them to clonal expansion, and then you have several secondary hits mutation that direct them to different phenotypes. So mirroring what happened in cancer that drive this biology, I think that can we go back to athero and think about, is it possible that the smooth muscle cell stem cell also undergoes initial mutations to drive clonal expansion, and there can be several subsequential hits that provide the heterogeneity of them later. So this is a hypothesis that I hypothesize the epigenetic and the somatic changes drive smooth muscle cell behavior in the plaque. There are several important questions I'm trying to answer in this study. So one thing is that I'll always ask, is it related to human from what we've studied from mouse? The other thing is that, is it chip or smooth muscle cell? So chip is a very hot topic recently that's been discovered by my senior colleague, C. Jeswa. So it says that the mutations in the leukocytes also attribute to atherosclerosis cardiovascular disease. So this form is called chip that indicate mutations unique to the leukocyte part can also contribute to atherogenesis. And I want to know that is it from the leukocytes or is intrinsic to the smooth muscle cell? Is it somatic mutations which happened in the long term, or is it epigenetic changes which can happen quickly? And of course, the trajectory of these changes in the smooth muscle cell clones is important. And by the time I mentioned this is a 5-year plan, I mean it's a 5-year collaboration plan within the Leduc plaque omics group that I'm fortunate to work with, and I have actually received the Junior Investigator Grant to initiate this project. So what we do is for this particular project to answer the mice and human question together is that the group with experts working on clinical bioinformatics and mouse models will pull our data together, the omics data together about human coronary plaque and also the mouse smooth muscle cell linear tracing plaque. And the bioinformatics will work on, again, alignment to see whether there are common mutations within the smooth muscle cells that's been driving smooth muscle cell crazy in the plaque. So my role in this project that I lead is to provide all the omics data based on the powerful mouse smooth muscle cell linear tracing models I have. It is important because if you scoop out the human lesion and you found a mutation, you don't know whether this is from leukocytes, endothelial cell, or the smooth muscle cell. So I am the only one who can provide the genome, the omics data to answer the chip or smooth muscle cell question, and also to provide a reference mutation that they can align later on the human lesions. And it's noted that at least for this expensive part of all the omics is covered by 2 grants that has been awarded to me. So here I show you at different layers how I would answer the question using smooth muscle linear tracing mice. So what I do is that for somatic mutations, I will of course isolating out leukocyte smooth muscle cell based on the linear tracing, and I will run the single cell RNA sequencing along with the whole genome exome sequencing. Here is actually one piece of real preliminary data showing here in the square. We have already identified several plaque unique variants that we'll working on later. And at the same time, to answer the question of epigenetic changes, I'm using the state-of-the-art single-cell attack sequencing to look at the chromatin accessibility into the smooth muscle cell. And by the unique thing about this pipeline that I show you here, established by my collaborator Howard Chang, is that by the time you isolate the DNA from smooth muscle cell, it's actually DNA from the genomic DNA and also the mitochondrial DNA. The mitochondrial DNA undergoes mutation 10 times faster than the genomic DNA. So it has a lot of mutation that might have been passenger mutations, but it's a great model for people to trace this mutation and to answer where does this cell come from in a way to track their clonal origin. So what this pipeline does is that when you do the single cell ATAC-seq, you not only find the epigenetic changes in the smooth muscle cell, but you will also take advantage of mitochondrial DNA mutation. You'll be able to lineage back to where does this smooth muscle cell come from to answer what are the epigenetic changes in this particular smooth muscle cell clones. So with the mouse and the human work has been ongoing at the same time, the next step is that we're trying to do the pathway analysis of the variants and the mutations we are looking for. And also to take it to the next step, we want to know that whether the mutation is meaningful or it's just a passenger mutations. And this can be done by using CRISPR-Cas9 to create the mutations in the in vitro culture model to look at how that drives smooth muscle cell behavior. And eventually we want to see is there some interesting therapeutic target that we can take out to treat a aberrant proliferation of smooth muscle cells. So for the second direction, which is to define the protein markers to identify human smooth muscle cell of different function, I have 2 major questions to answer. First of all, how do we know that this is a differentiated human smooth muscle cell? How do we find it? And then later on, how do we select the markers to represent the function of each of them to study what do they do in the plaque? So now if I remind you again, the StarNet co-expression model in which Johan also helped me how to identify the human equivalent SCAR1 like smooth muscle cell in human, which in human it doesn't express SCAR1, is that really by aligning the transcriptome from the single cell RNA-seq data, we'll be able to see that from the smooth muscle cell linear trace mice, this transcriptome profile can be mapped to human to see a similar transcriptome profile, then we can say that it's likely in the human equivalent of smooth muscle cell. And likely by the transcriptome matching, you will see that the human smooth muscle cell are also divided into several subtypes. And for each subtype, this is just an example. Actually, the transcriptome looks really close to each other. And very likely, you won't be able to pick up only one or two genes that's been highly expressed in each transcriptome to be able to represent that cluster. So what we do next is that we need to actually select a whole bunch of protein markers to represent each of the cluster. And these markers can be since highly expressed or somehow unique to that cluster, or since dramatically different or even down-regulated comparing to the other cluster. But at least we should at least use 4 or 5 markers to identify each of the subtypes. With this, then you really need the in situ multiplex imaging model in the next step to help you to dump all these markers together into the tissue section, into the plaque section, and to see where each smooth muscle cell phenotypes are. And by doing this, you not only identify the location of smooth muscle cell, you also link the transcriptome profile they represent into an in situ cellular context. In this way, you also actually link the pathway analysis from the transcriptome profile into the cell-cell contact with the cell interaction, cell microenvironment, and also tissue structure. Combining all this information together, then it's much easier for you to interpret the cell functions of what each cluster is doing in relation to ethogenesis. So this multiplex imaging system I want to mention about is called CODEX, which I would really love to work in Calgary with this CODEX. It's a Stanford innovative technique, and I'll show you here. I actually overlaid up to 40 markers, which are leukocyte markers, on top of the rainbow mice. And remember that the rainbow mice already have 4 colors on top of it. So this multiplex imaging system actually allow you to overlay 40 markers, and seriatically, if you design it properly, is unlimited markers on one tissue sections. And later on, it can also do a single cell segmentation. So we know that for each single cell, what marker they express. And later on, you can also even take the same section and overlay it with other imaging like the HE staining or immunofluorescence imaging. And with all this information together, if you are familiar with the technique CYTOF, which use a flow cytometry-like method that can probe multiple protein markers onto one cell, it is actually an in situ version of CYTOF that you can have a lot of information getting from just the one tissue section. So I have nothing to do with both companies, but I do think that the CODEX is a better spatial biology platform that we can set up in Calgary. So comparing to the 10x genomic technique, the CODEX is really targeting at things at the protein level. So I think that protein level can also show you a lot of the information related to translational mechanism study that a lot of people in Calgary are interested in. It's also easier to interpret the cell function from a protein level rather than the mRNA level. The other thing is that it's really single cell segmentation. So if you happen to have a tissue that cells are crunched together, it's easier to use CODEX to see at the single cell level what gene each cell express. And also, this platform is really flexible with archived FFPE samples, so you don't need to use a fresh frozen tissue. And if you also have very precious samples that you need to reuse it for histology, you can take out the same section after CODEX and apply it to other staining. So the good news about this is that the CODEX and 10x Genomics, both platforms are heavily involved in biomarker study for cancer and also immune disease. So it's the good news for a lot of people here who study chronic disease because the antibody panels is very immune heavy. So it's already commercially available, it's ready to use. But the bad news is that it doesn't have anything to do with cardiovascular disease so far. So spatial biology seems to be really underutilized for developing biomarkers and be applied to accelerate the study in cardiovascular disease. And here, I think that we really need a spatial biology platform to really leverage this technique to be able to find biomarkers and use it to find other diagnostic tools for cardiovascular disease. So here I talk about a little bit of my thoughts of my vision of how to do it. That this is actually definitely beyond 5 years. So you will have, each lab will have your unique interested omic profile you're looking at, and whether it's a mechanism or pathway you're looking for. But somehow, when you get the single cell suspension from this omics data, you obviously lose the cellular context to interpret its function. So if you combine it with spatial biology, such as CODEX or laser capture, you can actually link the omics into the cellular context. This will really help you to fit in the omics into the tissue structure, the microenvironment, and the cell-cell interaction to be able to interpret what does each cell do. And when you have that, you go back to the omics profile, then you choose your unique omic profile that can represent the cell function. And this is how you can generate new biomarkers. So here, I just want to list 2 examples that I'm really interested in looking at to have biomarkers in related to my previous study. So one thing is that from the omics data that I have and will have from aberrant proliferative smooth muscle cell, I want to see that well they are. So if they are here under the necrotic, it's definitely those are the harmful smooth muscle cells that we want to inhibit them from proliferation. And going back to the omics profile we have, we can identify that is there any biomarkers for this aberrant proliferative smooth muscle cell. So with that, we can actually screen the patient and to see that whether their patients are at high risk of disordered smooth muscle cell proliferation. And I think this applies both to atherosclerosis and aortic aneurysm disease. The second thing is that I'm interested in studying the metabolomics to find are there special metabolites secreted by the smooth muscle cell foam cells in which we can customize it to use it as contrast reagent. So later on, maybe we can image smooth muscle cell foam cell better in this way that when we design lipid-lowering therapy to treat patients when they are in clinical trial and try to make the decision whether to go or not to go from phase 2 to phase 3, we have a pharmacodynamic marker to evaluate how effective they are to reduce smooth muscle cell foam cells. And sometimes this decision making in pharmaceutical sciences really costs you $1 billion. So these are the 2 things that I think that I can actually work with experts at Calgary who are interested in immunology, who are interested in vascular diagnostic platform, and who might be also interested in also building a spatial platform for cardiovascular disease in the future. So here, I want to thank everyone who has been, who I have been fortunate to work with. They're the people who really opened my eyes and helped me to look into different avenues that I in the cardiovascular disease, and really encouraged me in my career to help me bridging into the next step. And now I'm happy to take any question.
Great, thank you so much. What an outstanding talk, and congratulations on all of your work to date. Absolutely fascinating. And well done. So we do have a number of questions, which I'm going to moderate. So we're having people send in their questions through the chat line, and I'll ask them on behalf of the submitters. So the first question is this one. It's from Sarah Child. She says, I'm curious about your proposal to use exome sequencing, if I interpreted this correctly, to look for somaticism. Wouldn't whole genomes be better? Calgary has tremendous expertise in sequencing for somatic variant detection.
Yes, yes. Actually, the whole genome exome sequencing is something that we have just proposed to do, and this is actually a conversation that I have been going on between me and a bioinformatics person, in which he also suggested, hey, Ying, I think you should do a whole genome sequencing rather than the whole exome sequencing, because you can get a lot of information that you may miss out from the exosome. And I agree. So we have been working on to look at the pro and con of both methods. I think the whole exome sequencing do have a little bit more reading depth compared to the whole genome sequencing. But I think we are open at ideas of whether it's a whole genome sequencing or whole exome sequencing. Actually, this part has just started, and we are actually trying to decide which one to take. So I'm happy to getting advices from people at Calgary. So it seems like our bioinformatics person is definitely has his alliance over here.
Excellent. Okay, this is maybe part two of the question from Sarah Childs. She says, I'm really excited by your single cell RNA sequencing of smooth muscle cells, particularly looking for a smooth muscle cell stem cell. How do you see that profile relating to embryonic smooth muscle cells? Any idea if they have an overlap in gene expression profile? Again, there is expertise in my lab in this area, but in fish developmental vascular smooth muscle cells and their precursors.
So can I just ask you to clarify that question? So when you mentioned about embryonic smooth muscle cell stem cell, do you mean that in the human part or in the mouse part, or it's in general?
Why don't you answer that? Yeah, go ahead, Sarah. It'd be easier.
Okay. Yeah, no, it would be in mouse, but in general, I mean, often stem cells, in cancer, whatever, take on embryonic properties. So the question was, do you think there would be any value in comparing that to an embryonic profile? It was just a simple question.
Oh yeah, I think we found that a lot of the embryonic stem cell, we found that it's located actually in the adventitial part. Actually, I got a lot of questions saying that, do you think that it's possible that this embryonic type of residential smooth muscle cell stem cell resided in adventitial, and then the skull-one smooth muscle cell you've seen here actually is moving from the adventitial into the intima rather than having them cooking by themselves in the intima itself. So the answer is that no, we haven't really looked at the embryonic stem cell part and comparing it to the skull-one smooth muscle cell part. But at least that we show that in the normal blood vessel, we didn't see any of the skull-one smooth muscle cell. But I think that during embryonic, we don't know if that's happening, if there is any skull-one expressing smooth muscle cell during embryonic formation.
Great. This next question is from Dr. Morley Hollenberg. It says, in your schemes, roles were proposed for proteinases, examples, MMPs targeting matrix and proteinases of the complement system to generate C3. So the question is, do proteinases or their inhibitors surface in any of your transcriptomic analysis?
That I have to go back and to look at the data of the original pathway analysis. I do remember that the most significant pathway that has been upregulated in the SCA1 transcriptome profile is there are a lot of the complement pathway in it, and along there is also a lot of the MMP have been upregulated in the transcriptome profile. But with that specific protein, I don't have the memory of seeing it being very on top.
Okay, thank you. This next question comes from Professor Ed O'Brien. He says, what is the evidence for aberrant proliferation of smooth muscle cells in human arteries? What is the degree of proliferation?
In human artery, actually, I don't know how fast it proliferates because it's hard to trace proliferation in human vessels. So I'm a mouse doctor, but at least I can answer the question from the perspective of mouse. So there I remember that when I did experiment, you can inject into you to label what are the proliferative smooth muscle cell in the plaque. And then when I isolated the plaque, it seems like their proliferation rate in mice is not that fast. So within 24 hours, I've only seen about 3% of smooth muscle cell in the plaque being proliferation, and that's undergoing proliferation. And a lot of them are actually SCAL1 positive smooth muscle cell. That's one of the data I didn't show to show that SCAL1 smooth muscle cell are highly proliferative. But of course, in human, I think depending on what disease you are trying to look at, comparing to, so human osteogenesis is a long-term process that can happen within decades. I would say that the proliferation in the long-term osteogenesis maybe is not that significantly as fast as really a tumor cell growing on the blood vessel, because that takes decades. But in terms of aortic aneurysm, which may happen pretty fast, and it's said that in this disease, smooth muscle cell play more of a proliferative role compared to osteogenesis. So I think within that disease context, I would expect more smooth muscle cell proliferation in aneurysm disease.
Okay, this next question has a number of parts, and I'll read it, and I'm happy to read it again for you. In your previous data, you showed that smooth muscle cells upregulate C3. Following this, you build your model of enhanced efferocytosis. However, how does C3 get cleaved? C3 is part of a proteolytic cascade with many different complement regulators and convertases that need to be present. Follow-up, there is 1 milligrams per mil present in the blood. How does the little local production contribute?
So that's a $1 million question that the reviewer also asks is how do you get C3 cleaved in the lesion? So my way to answer this is I actually went back and did multiple staining about in the lesion, and I found that a lot of the cleavage is actually happening. So when C3 is cleaved, and also there are other mechanisms to help to regulate the downstream part. So what I see is that a lot of the cleavage product, the end product will be generating the C5b-9. So when I stain the lesion with C5b-9, a lot of the C5b-9 is actually enriched in the luminal part that's close to the lumen. So I think that a lot of the factors that help to trigger the later on cascade after C3 and trigger C3 cleavage is coming from both the lesion part and also from the blood product itself. So that's why you see the cascade end product has been really close to the lumen. And in the deep intima, I see that if there is a factor can infiltrate from here deep down inside, then it's likely that the C3 will also get cleaved in the deep intima. Because remember that in the plaque, because of the endothelial cell has already lost its function to being a barrier, a lot of the factors from the blood can infiltrate through the lumen into the deep intima. So that's how I think that C3 can be cleaved.
Excellent. Someone had already mentioned the adventitia, and that was the question that I wanted to ask you about. Is, you know, my lab is particularly interested in the formation of aneurysms, and we've been exploring to some extent the role of the adventitia, and there is some interesting data to show some relationship between inflammation and immune cells and fibroblasts in the adventitia as mediating that disease. And you did mention that you had maybe one publication in that area. I'm just wondering your thoughts on the role of immune cells and fibroblasts from the adventitia, maybe even in atherosclerosis.
So the immune part is actually not my strong part, but I would say that even though I appreciate that smooth muscle cell is a residual risk factor, but I'm not objecting that immune cell play a very fundamental role to initiate the disease. Actually, there's a huge debate whether atherosclerosis or aneurysm is a disease that's triggered by macrophages or smooth muscle cell. I think it is a collaboration of both. And regarding to your specific question about fibroblasts and the immune cell in the context of aortic aneurysm, I think the way to learn it is that with immune cells, I think that during aneurysm, my feeling that it's more of a smooth muscle proliferation disease. But how the smooth muscle cell get proliferate over there, it has to be some trigger. And I think that the trigger might be cytokines released from the immune cells, and that as a part of an immune response. And regarding the fibroblasts, I think fibroblasts is also an area that has been really hard to study because fibroblasts is just so close to other cells. It undergoes phenotypic changes dramatically also. So I don't know particularly about how fibroblasts can bridge into aortic aneurysm, but I think that based on my study, when I isolate the plaque and try to culture them, fibroblasts is definitely a very important part that support the growth of smooth muscle cell. When I take out this fibroblasts and just saw the smooth muscle cell, my ex vivo culture doesn't go very well. So definitely fibroblasts is provide a nutrition layer for the smooth muscle cell, and that has been highly proliferated, and also it can sequester maybe a lot of growth factor like VEGF to provide like an incubator for the cell to grow in aneurysm disease. That's what I think.
Excellent. Okay, this next question comes from Professor Koobs. He says that he's just purchased a multiplex ion beam imaging system from Stanford. How does it compare to the Kodak system?
So I think the cost is mainly at the cost. So maybe it's also a very new thing to everybody. But what maybe has is that I heard some inside news is that depending on how comfortable you are with the system, it's less stable, somehow a little bit less stable than Kodaks. And also, if you look at maybe it's a really in situ version of Cytov, though I compare it before like a Kodaks, but Kodaks is really still antibody-based level. It's more of a low-hanging fruits that people are more comfortable, like beginners can use this Kodaks system to generate their own conjugated antibody. Once you figure out the antibody, and then you can apply it just as an imaging scanning system. Whereas I heard that maybe has a little bit less stability, and also is a little, sometimes I heard that it's a little tricky and more expensive because considering the antibody you are conjugated are all of the expensive metals that you use. Like, yeah, I think that's based on the cost of the antibody and also the system. Someone just told me that even for the platform that you need to load the sample with maybe is that you need to have like a heavy metal or gold metal that for you to hold the tissue, which you don't have to do that with Kodak. So Kodak is definitely cheaper, but with resolution, I definitely think that maybe it's a way more advanced than Kodaks. First thing is that it also can zoom into intracellular structure like the cellular organelles. So it's really not only single cell resolution, but intracellular resolution. Second thing that the advantage of that is that it remove a lot of the background noises and a lot of the spectrum overlap you may have seen with Kodaks and other antibody-based method. Is that these heavy metals they conjugated, it's harder to have spectrum overlaps, and also for study FFP sample, usually by imaging you get a lot of background noises, whereas with maybe because of the analyzing, it reduced the background noises a lot because those heavy metals conjugated antibody will exist in your signal, but your tissue background will not have those special metals exist. So I think that's the two major differences between MIBI and Kodaks. So I personally haven't worked with MIBI before, but I heard a lot of inside news from people in Gary Nolan's lab.
Excellent. Okay, this next question comes from Professor Andy Braun. He says, Dr. Wang, do you know if vascular smooth muscle cell foam cell formation / proliferation is reversible once the HFD is ceased or statins are introduced?
Once the HNDs, so I missed the
Yeah,
later
so
half of the question.
do you, and Andy, feel free to pipe in if we're not transmitting your question properly. He says, do you know if VSMC foam cell formation is reversible once the HFD is ceased or statins are introduced?
Oh, I
High-fat
see.
diet.
Yeah, I see.
High-fat diet.
Yeah, I see the question. This is, so first of all, if we just remove the high-fat diet from this mouse, they don't have lesion regression. So we get this question from one of the reviewers saying that, oh, can you just remove the high-fat diet and see whether you can see plaque regression in this mouse? So we didn't do it because we have I have discussed with some collaborators who have done the experiment, like I think Edward Fisher, who's the expert in microphage regression and plaque regression. He said he emphasized that at least in the mouse model, for you to see regression, you would at least need to transplant that blood vessel all the way to non-apple knockout mice, normal mice, and simply by changing the diet will not help to see the regression effect. And with that, so I don't know if removing the diet will help, but giving statin is another thing, is that mice seem to be not responding to statin therapy very well. And that, I think there's some fundamental differences between mouse and human models. But I do think that in human, I hypothesize that because this smooth muscle cell is like a dead trap for cholesterol. If you lower down the LDL level, it may help with reducing the smooth muscle cell foam cell formation. But once it's formed, I think we need to target at how to properly upregulate the ABCA1 level on this cell to extract, to vacuum out the cholesterol from these cells. I don't know that by lowering the LDL would have a significant beneficial effect on human related to removing smooth muscle cell foam cells. But that's actually one of the targets that Gordon Francis' lab is working on, is to target at the metabolism pathway in the smooth muscle cell. So the answer is no, I don't know if studying would help, but definitely if we can linear trace smooth muscle cell in the human plaque later, it will be a really interesting story to see whether by archive the sample, people having having smooth muscle cell foam cell and people also have had statin therapy before. I expect that maybe a lot of the remaining foam cell population could be smooth muscle cell foam cell. If we can find that evidence, then it's telling you that the statin may be not, the smooth muscle cell foam cell may not respond very well for statin therapy.
Okay, excellent. Outstanding work. So that ends the question round. So we want to thank you again for this public lecture. Really outstanding job, and we look forward to connecting with you later today.
Thank you very much. And I see you later today.
Yep, take care, everybody.
Thank you.